we'll talk about catalysis as well. But we'll talk about catalysis after we look at derivatization. And the derivatization will involve porphyrin derivatives. So we'll have a chance to think about porphyrins, okay? So I just have a quick question. How many faculty and students are out there listening? Are there mainly students or mainly professors, or what kind of breakdown do we have?
Half and half. Around 50% is lecturer and 50% student.
Oh, okay. So I guess I'm gonna try to aim this a little bit more for the beginning graduate student. And then let's see how that works. So in the first part of my talk, if there's a problem with this screen share, let me know. Otherwise, so we're on a title slide that begins cellular and catalytic, blah, blah, blah. Okay, so let me just go, actually, the second slide, I just wanted to recap, and many thanks for the visitation. It was 6 years ago, and I had a wonderful time down in Indonesia. It was my first time there. And I had a chance to meet the student who was going to join my lab at that time, Tesla Yudhishthira. You can see down, oh, by the way, can you see the cursor? When I move the cursor around, can you see the cursor?
No.
A little triangle? Okay, so I guess I won't rely on this pointer. So, okay, well, yeah, in the blue box at the bottom, yeah, I just wanted to first thank Adi, and then also Tesla has gone on to bigger and better things. He's a postdoc over at the University of Strasbourg. Now I have a current Indonesian student, Arik, who's with me. I'm really happy with Arik, so we do a lot of good work together. In the first part of my talk, I'll be talking about porphyrins that were part of a project that were kind of between my lab and Professor Gross's lab. He works at Technion in Israel, and there's a lot of great research going on in his laboratory regarding porphyrins. So I wanted to use this opportunity to talk about photocatalysis, and the synthetic work was done in his laboratory, but a lot of analysis and writing and all sorts of other considerations were done kind of conceptually with a team. So if Zev had a great Chinese student named Zhuan, so Zhuan was working there during my year in Israel, which was in 2017. I also wanted to thank the IBS of Korea and also KAIST. So these are this is sort of my acknowledgement slide before I forget, because I sometimes run out of time and then I forget. Sometimes I honestly forget to thank the people that are so important. So yeah, just a really quick visual as to where we are. Daejeon, upper left. Daejeon is south of Seoul. It's in the Korean Peninsula, and there's some interesting nightlife scenes, and there's this view of the campus on the bottom of the slide. So these are just pictures of where we are, where we're working and living, and I wanted to also mention that as chemists, we're constantly kind of in pursuit of something to do, something important to do, and that has brought me more and more into biology. I was trained basically as an organometallic chemist, and then the question is, well, how do we save the world? How do we do? How do we think about the next billion-dollar idea, et cetera, et cetera? Well, I guess the answer is always in like these baby steps, thinking about things, very logically and stepwise. And I've been very interested in thinking about dementia and the aging process. Oxidative stress, right, is one of the causative factors. Obviously, people talk about proteins and also metal ions, but if you ask yourself, what is the normal cause of what is the cause of dementia or sporadic dementia? Then we need to find that out. So the chemists will come to will be part of the team that's involved for sure, but obviously there's a lot of biology. So, and on the right side, just a couple of pictures of people that passed away with dementia or related to illnesses in the entertainment and sports world. So sometimes people put the picture of Ronald Reagan up there or something, and some conferences we've been involved with to try to bring people together to think about these problems over the years. We try to have them every 2 years. And as an inorganic chemist, there's a big breakdown of research, and there's what's called the main group. That's the P block, and I think we've all seen and thought about the periodic table at some point. The students are probably a little intimidated by the periodic table, but we can think about group 16 and group 15 in this talk. So the chalcogenides, that would be the group 16, and the so-called pnictogens are group 15, right? That's nitrogen, phosphorus, arsenic, and so forth. So we'll get a kind of a different picture of phosphorus chemistry after today's research. And in the background, there's the web page to a journal, and the journal title is Main Group Chemistry. So in the future, I'll be heavily involved with this journal. So I'm going to try to bring kind of, in my own research, the kind of the reasonings of biologists, try to make sense out of what we can do with main group chemistry in biology, or also just generally in catalysis and environmental chemistry and so forth. So on the left, we can think of a bottom-up approach. On the right, we can think about a top-down approach. And either way, we kind of get to a product, and the product is, they are kind of novel compounds. I suppose they could be known molecules where you have some, you know what you want to get and you get there. But in our case, we find kind of a joy and an importance in just trying to go with some kind of new or kind of unknown system that we're not expecting much from, perhaps, but we try to make the most out of it. You can think about, again, atomic positions and metal ion coordination and so forth. There's a lot of considerations, and we can get to a point where the molecule can interact with ROS or metals or even proteins. So if we think about molecular probing, these are kind of some concepts: selectivity, reversibility, and even connectivity between the probes. So in the first part of my talk, I wanted to discuss, try to get over to some findings relating to photocatalysis. And for the students, this is kind of a nice summary of skeletons and numbering. So if you have a macrocycle, these are three macrocycles. On the left, you have the porphyrin. The porphyrin is a regular and relatively symmetrical, or quite symmetrical macrocycle, and it has, if you look in the core, there's 4 nitrogens and 2 protons, and on the right side you have corin that has 1 proton, and then you notice that in the skeleton there's actually 1 fewer carbons. Well, in the chiral system in the middle, the chiral system possesses aromaticity, which porphyrin does, and it also possesses the same kind of carbon skeleton that the corin does. So we're kind of looking at a combination of the 2 systems in one, and we can do a lot of things. This is a popular macrocycle. It's more difficult to work with than a porphyrin, but it's more, I think we find it's more rewarding, and there's a lot of effort that's ongoing. So this is what I started doing when I first started at KAIST. And this is what Professor Zev Gross has been doing for many years. He's the world's expert, or one of the world's experts in chirols, one of the few people that's really focused singularly on chirol systems. So I'd say he's the #1 chirol person in the world. Okay, so well, another aspect of main group is you can actually, instead of thinking about some central element, you can think about some substituent. Later in the talk, we'll look at selenium as like some derivatable, some kind of side chain kind of approach that we're looking at. But here we'll look at the trifluoromethyl group. Okay, so we're going to take the chirol macrocycle, and we're going to take the phosphorus, for example, the phosphorus as the central element. Now, you might be surprised about phosphorus. Actually, phosphorus can sit in the middle of the macrocycle. Instead of a metal, you can actually have a main group element sitting in there. So, and then on top of that, we're going to add a couple of axial ligands to the phosphorus. We're going to say, let's put fluorine up and down. So there's like a PF2 unit there. And then on the outside, if we go back to if you can mentally think about the macrocycle, right, there's the outside of the macrocycle that has the CH groups. Well, we're going to substitute some of those CH groups with CF3 groups, and we're going to try to make sense out of that. So we're making isomers. Obviously, if you're adding substituents, you can add one or two or multiple, and then they can be located at different places. Now, practically speaking, you can do a lot by NMR, for example, but sometimes there's no substitute for a nice X-ray diffraction study where you can find, you can locate some particular substituents and be sure where they are. Sometimes it's not clear by NMR. So the structural characterization was something that we undertook. Also electrochemistry, and then we can start to formulate one or multiple hypotheses. And the MO molecular orbital diagrams are able to be obtained through calculations. So we'll look a bit at, and that was actually done by my colleague Mookie Bake in our department. So there's a lot to discuss with MO calculations. And finally, photocatalysis. So the photocatalysis, you will see some of it involves taking a chloride and then using the phosphorus corol as a photosensitizer. So the photosensitizer will interact with oxygen and form singlet oxygen, and then that singlet oxygen is thought to then interact with chloride or bromide, and it'll change chloride or bromide to chlorine Cl2 or bromine Br2. Now those Cl2 and Br2 molecules will then in turn undergo reaction with some substrate. So what you need to have is some light, and you need a photosensitizer, and you need oxygen, and you need some chloride, and you need some substrate, and then you can think about doing catalysis. Yeah, so before I go into all of our research, I encourage you to think about maybe in your own laboratory, if you have something that's a pi delocalized system, if it's a new molecule, you can study photocatalysis. So what you need is like some light source, again, obviously, and some simple ingredients, and you can test your own molecules. So I encourage you to do that. We went pretty far into this derivatization study, but there's a lot you can do in a simple way. So this is the citation. Slide 11. So we're all on slide 11, hopefully. And the title is The Synthetic Roots, etc. So yeah, we're showing you the molecules that we synthesized. And some of the yields are not great, but you know, with synthetic chemistry, sometimes getting something pure and isolated, even though it's 7% or even less, is a nice thing to have, and you can study the molecule quite clearly. So along the top, we have compounds 1A, B, C, D. And in the bottom, near the bottom, we have compounds 2, 3, 4, 5. I'm not going to go in. I could probably spend a lot of time talking about synthesis, but why don't we just mention that these have all been made and characterized and isolated and characterized. And we're going to look at the differences soon, especially regarding the number of CF3 groups that have been added. We can also compare with other substituents such as bromine, right? So bromine, this pentabromo system on the left, was used to synthesize the CF3 groups, the penta CF3 group system. And you can compare this penta. Apparently, when you try to brominate the phosphorus corol, you can't go all the way. So you can brominate 4 or 5 or 6 positions maybe, but you can't brominate all 8. But for other systems, you can brominate quite easily. So there's something going on with this system. You can see that the PF2 unit in the middle, right, the phosphorus in the middle, I always think of that as like an electron withdrawing group, like internally in the system. For the gallium, it's a much more electron-rich system, and it's a gallium-3. It's not a gallium. So P is actually 5+ formally, gallium is 3. So we're kind of comparing apples and oranges. But what you can do is just say, oh, well, this one's highly brominated, and how does it compare in catalysis, for example? So even though you don't have an exact comparison, sometimes you can use something as a standard. And this octobromo gallium system has been used as a kind of standard in the literature. Okay, well, if we look at the absorption spectra of the 1, 2, 3, 4, 5 substituted corollas, this is where we have 1 CF3 group, or 2, or 3, or 4, or 5. What we find is we have a very similar looking signature. But for the students, I wanted to mention that whenever you're looking at a coroll or porphyrin type of system, you're going to see the Soret band, that's S-O-R-E-T, Soret band on the left, and you have the Q band on the right. So the Soret band and the Q band are two very prominent features in in porphyrinoid chemistry. I think it was probably studied 15 or 20 years ago. I mentioned that phosphorus corollas are highly fluorescent. Now, even the gallium is like a heavy atom. As you start to put heavy atoms into a system, you're going to start to encourage the singleted triplet. It's the intersystem crossing mechanism. Whether the heavy atom is in the middle or kind of on the outside, it's affecting the pi delocalized system significantly. But for phosphorus, you don't really have that issue. So you can have highly fluorescent compounds. But again, if we look at these closely related systems with whether the CF3 is on one carbon or the other, we have a similar kind of signature for the fluorescence. But the point here really is the fact that we do have fluorescence. So the fluorescence exists. And I wanted to mention just a little bit about characterization, because we're very proud to be able to isolate these different isomers, whether we have a CF3 group on position, technically speaking, position 1, 2, or 7, or 8. These are all reflected in the NMR species. So on the top we have 1A. Well, 1A, on the top we have the non-substituted system. And what you see are 4 signals. And then the next one down, the blue one, is when you add 1 CF3 group. And then what you lose is the symmetry. So you have a kind of nominal C2V symmetry, and then you lose it to having just CS molecular symmetry. And that's also true for 1BCD. Okay, so we're seeing purity, we're seeing loss of symmetry. You can do a lot with fluorine NMR. Hopefully, as a student, you have some, if you're interested in synthesis, you have a chance to do a lot of NMR or some NMR. If you're having trouble getting spectra, you can always collaborate with larger universities, or where I don't know where you're working. Maybe you're all from different universities. So when you're dealing with NMR, right, you have to consider what a signal really is. And then how many you have, and it tells you something about symmetry and so forth. And fluorine, if you ever have a fluorine or a phosphorus in your molecule, you can do a lot of things with that in understanding the structure and purity and so forth. So for the unsubstituted system, the top purple line, this is really showing kind of the same spectrum, but just different regions. So on the left, we're seeing the PF2 region. And in the middle, we're seeing where the CF3 region is. So the first one is kind of the unsubstituted system. So you don't have any CF3 group until you get down to 1A, B, C, D. And then on the right, you have what we're looking at is the C6F5 aryl groups that are in the 5, 10, and 15 positions. And we're just looking at the most remote fluorine. So that's where you have the para position. And in the unsubstituted system, you have a symmetry. that is lost by the other substitutions. So this is I guess the bottom line here is, if you have a pure compound with a fluorine, try to spend time and think about what it's telling you. So you can see the large coupling in the PF2 signal. You see the large coupling. It's actually just one signal, but you have a wide, like a 900 hertz difference. Okay, so another trick with NMR is you can actually remove the coupling, the phosphorus coupling, and in the proton NMR. So this is the protein NMR now that's become simplified because we're subtracting out the influence that the phosphorus was having in coupling with the different species. So now, if you look down at the 1ABCD spectra, you see a singlet. So that singlet is where that's the neighbor of the CF3 group, right? There's kind of there's like 4 quadrants where you have CH resonances. If you look at where the CF3 group is, the next door neighbor, right, is a CH, and then the CH, if you subtract out the phosphorus coupling, appears as a singlet in this experiment, in this NMR experiment. So you can really appreciate where the CH group is. So if you have a chance to take NMR of porphyrins, try to go as far as you can in understanding the coupling, and it's for your own training. And you can learn a lot from the literature, but it's also good for you to try to learn to do things on your own. Having said that, sometimes a university will have an NMR technician. So what that means is you give the sample to somebody and they do it, and you never see anything about the process. So I encourage you to try to go, if you're interested in characterization or synthesis, try to see how things are done by NMR or by any technique that is done by a technician. Try to just learn more from the technicians. In this slide, we've taken the structural PF distance on the x-axis and correlated that with the coupling, the PF coupling constant on the left and on the right. We've thought about just generally the chemical shift. Try to make some correlation between structure and the NMR. Now here we have, importantly, electrochemistry. So bottom line is the one that's pentasubstituted, that means 5 CF3 groups, was found to be special, really special. So, and then we can see why it's different by DFT, theoretical calculations. But basically, you can see as you add 1, as you go from the purple, the 1, 2, 3, 4, 5, you go from the purple to the green to the blue to the red to the black. You're kind of going to the right side. You're making it harder to oxidize. And there's a plot that's showing that the effect of these number of substituents on the reduction potential, for example. So you see, and then that slope is telling you that if you add just a couple or 3 or 4, then you're really having a big effect in your system. So this slide is the bottom line here, saying, the CF3 group packs a punch. It's really has a strong influence, and that was Professor Gross's general idea, and it's a really nice substituent to consider, to think about. So that you can compare it with, you want to kind of add a couple of substituents that really have a big effect, because you run out of places, right? There's only 8 CH groups that you can substitute. So if the effect is not great, you keep substituting and substituting. You kind of max out your system, and then you haven't really got to where you want to get to. Right. So the idea is let's use something, a substituent that's more aggressive. Obviously, the characterization, the synthesis is more difficult, but you can get to something that's much more interesting to study, and the withdrawing capacity of CF3 is really evident through this data. Okay, a couple of plots. So this is now on the y-axis. We have the coupling constant. It's the same on both sides. And on the x-axis, we have the number of CF3 groups on the right, on the left rather. And we're seeing a nice linear correlation. And then on the right side, we're actually saying coupling as a function of reduction potential. So we're also seeing some linear agreement there. So this is setting us up to think about the structure itself. Oh, there's a few. Yeah. So the ChemDraw images are the compounds that we were able to crystallize. Okay. Now we isolated everything that we discussed, but it's really a special thing when you can have a crystal structure of your compound, because not only are you kind of guaranteeing where that substituent is, you're also getting some more information, and that additional information may not be immediately clear, because if you look on the right side, these are the structures: 1B, 1C, 2, 5. Okay, and they're correlating with how many 1B, 1C. They have 1 CF3 group, 2 has 2 CF3 groups, and 5 has 5 CF3 groups. You can see that the macrocycle is doing something weird, right? It's doing something weird. So what is it? How do we understand what it's doing? Well, number 5 is distorted. Okay, so how do we understand distortion? Well, you need to have some system in order to understand the distortion. So the systems are taken from, I think, as I remember, taken from the Porphyrin literature, saddling and ruffling. Okay, saddling is where you have like the saddle point saddle, if you've ever seen a saddle on a horse or something like that, you know the shape that we're talking about, saddle points. So the saddling is defined as on the left, and the ruffling is defined on the right. Obviously, a Corolla is more constrained than a Porphyrin because you have slightly fewer more atoms, but this is what's happening. So this is how we can define what we're talking about. If you see some distortion, how do we understand the distortion, right? You have to kind of quantify it, and then you can compare between systems. Okay, maybe I can see that I should probably speed up a little bit. Let me hop over to this interesting slide. Now this is just the normal PF2 system without any trifluoro methyl groups, and you see four diagrams. You see the LUMO +1, and the HOMO -1. Well, these are the famous Guterman 4 orbitals, right? So what we have are these orbitals that are the frontier orbitals, okay? And these frontier orbitals are what's gonna help us derive the spectra that we see. The fact that you go from one, you have an option of HOMO or HOMO -1, means that the electron can be promoted from HOMO or HOMO -1. And the fact that you have LUMO +1, we always do, but the fact that you're seeing the pictures, you're seeing this intimately here. LUMO and LUMO +1, there's two like receiving orbitals. So this kind of gives rises to four different transitions. So that's what's if we think about the absorption spectra, what I talked about, the fact that you have a Sorey band and you have a Q band. Well, that's coming from this information, right? These are the orbitals that are involved, and then as you add CF3 groups, you're going to change the nature of these orbitals. So this is an output of geometry optimization. And it's an output of what's called an FMO analysis. So that's known as the frontier molecular orbital analysis. If you go over to the number 5, you can see how all of the frontier orbitals have kind of decreased over the course of the plot. We also had a chance to look at number 8, which is a theoretically octa substituted system. In other words, the crazy one, the one we could never make. And that also that shows a real decrease in the orbitals. So these frontier orbitals are all kind of going down, and that's the sort of the beauty of the import. That's how that rationalizes what we see in the CV spectrum. Just a visual on the calculated orbitals and the same pictures of the MO orbitals, MO diagrams we just saw. Now, for catalysis, as I was trying to say in words earlier, what you start off with is a hydrohalic acid, HX. It could be HCl, it could be HBr, it could be even HF. And what you can do is that's the source of the halide. Okay, the halide is then going to be attacked by what we think is singlet oxygen. Although there's a lot of, if we think about ROS, there's a lot of intricacies that come into play. But a simple thing to discuss is to know that our probe, our photosensitizer, is receiving the available light energy, and is transmitting it to oxygen, which is going from the triplet, the ground state triplet oxygen to singlet oxygen, and then we think that's combining, that's hitting the bromide and making it bromine. So we're changing Br minus to Br2. Or, for that matter, we could change Cl minus to Cl2, and so etc. Now, we had a chance to look at particular wavelengths. You can see on the upper right, 400 or 450. And sometimes the reviewers will complain, why did you use exactly 400 or 450? Because the maxima, like the absorption maxima, are slightly different from that. But we managed this, and we just try to keep everything as consistent as possible. And what you see, if we compare number 5 down on the bottom, we can see what we get when we add HCl and we add light and we add our photosensitizer. And what we end up doing when we're studying phenol is we're getting a substitution. Either the substitution is on the para position or on the ortho position, so OP. Okay. So, and we just combine both of those, the concentration of both of those, and make a plot, and we can see that our phosphorus Carol is really doing a good job, much better than the gallium octobromo system. That's where instead of having 5 CF3 groups, you have actually 8 bromines around the outside. And you can also compare with the pentabromide. Okay, so we're seeing that the withdrawing effect, the effect of the substituent, the CF3 group, is much stronger than for the bromine when we combine it with the gallium, or even with phosphorus, when phosphorus is then derivatized with bromides. So we looked at different wavelengths. You can see on the left, on the bottom left, we used 400 nanometer light, and in the middle we used 450 nanometer light. That's a less aggressive wavelength. So this is really helping us appreciate what's possible, and it's more realistic relating to solar energy, but it's also important in health sciences, because there's a lot of activity where you're looking at in a microscopic way, microscopy, where you're looking at fluorescence, and you're looking at cell, and you're looking at cancer cells, or you can think about even PDT, which is the photodynamic therapy approach. And how can we adapt photodynamic therapy to do something good in the realm of neurodegenerative disease research, for example? So all of these kind of applied questions come out of this basic research, these aspects. Here's just a glimpse at what we do to try to measure the amount of phenol and the phenol derivatives. We use a GC, that's gas chromatography. So this is like a chromatogram, that one of them you would consider looking at and measuring. Okay, oh my gosh. So I'm at about 35 minutes. There's a lot more we did with catalysis. Yeah, so the next very simply, the next paper dealt with looking at gallium and aluminum corolles. And we looked at what's possible with the phenol system. And in this case, we also observed the benzaldehyde formation. And that was an interesting result because that does not involve, the bromine, right? We're talking about some other reactive oxygen species, right? That's hitting the methyl group. And there could be more than one option for that product, the formation of that product. So that was something that kept us busy, and we had a chance to compare with the CF3 groups that were involved. In the paper after that, we looked at kind of having two sets of substituents in the beta positions. You can see that there's CF3 groups, and then you can see there's also on the top, there's bromine groups. So we referred to this as like a hetero multi-functionalization. So we had two important sets of beta ligands, and then we sought to find out what good that is. So obviously, if you're adding bromines, you're going to have a red shifting of the system, and we also observed phosphorescence. So we were able to achieve room temperature phosphorescence with this system, and then we also did do some catalysis, as shown here on this slide, where again we used phenol, and we studied the various differences between gallium and aluminum. Oh no, I think in this case we didn't look at aluminum, just gallium in this case. Okay, and then in another paper we had a chance to look at whether one or two or three methyls, right? So what happens if we have more than one methyl group? Well, we find that we can change a methyl group, an aryl methyl group, into a CHO group. Okay, so and we found, long story short, that the phosphorus corol that has five, let's see, PF, the phosphorus corol that has four, let's see, oh, it's here, yeah, it's called 4P. It has 4 CF3 groups. That was the one that had the highest activity. And that's shown in the yellow bar in the graphs. It's shown in the yellow bar. Whether we're dealing with toluene, or whether we're dealing with xylene or mesitylene. So in your research, right, when you're doing a study, you can try to think about a systematic series of compounds. And we had a chance to add the I3-, which is going to trap the singlet oxygen. So we had a chance to try to look into the mechanism of this, and this was probably the most interesting paper. And there's a little bit more information from the paper here. Okay, so conclusions about this part of the talk. Synthetically, we were not able to achieve every single compound imaginable, but the 5 CF3 groups had a strong effect. They were optimal. The P derivatization seems to be kind of a special kind of species. Also, it's very stable. It's kind of wonderful to work with. So if you're thinking about which porphyrin or corol to use, if you really want to go into that direction, the research direction, I encourage you, if you're interested, try to use phosphorus and see what you can get. You can try it for a while, but there's a problem when you have the hydroxys, right? Because there's hydrolysis. So if you try to get over to the PF2 unit as soon as you can, that will help your research. That will help the synthesis. The macrocycle deformation was a factor. We tried to quantify that using the puckering, like the two ways of mathematically determining whether we have saddling or so-called what's the other one? I'm forgetting it now. But yeah, there's the saddling, and then there's the other kind of deformation. So then we can think about not only substitution, but also a deformation and an activity relationship. So you're kind of starting to twist the whole entire macrocycle. We were successful in our photocatalysis, and we looked at I guess a reasonable number of substrates. They all tended to be aromatic. The aliphatic ones are obviously less reactive, but it was an interesting study for sure. And we had a chance to compare with aluminum, gallium, and gold, and even cobalt systems. Okay, so I'm at 41 minutes now. If I can take 5 more minutes, is that okay?
It's okay. Okay.
Okay, thank you very much. Yeah, I'll take 5 more minutes and go from the phosphorus section to a sulfur or selenium section. I think you'll find this interesting. So biophiles are a kind of natural constituents to proteins in biology, and they're known. The glutathione, for example, on the bottom is known as the famous antioxidant, and in our body. And if you think about the fact that the SH can form the disulfide linkage with itself, for example, then you can kind of determine in any biological system, in any like mammalian system, for example, how much glutathione versus how much reduced glutathione versus how much oxidized glutathione there exists. And that kind of gives you an indicator. It's like a barometer for how much oxidative stress there might be. And it is almost always highly reduced in character, in biology, in reality. So we want to try to think of a probe system. So we're thinking about how these biophiles can react with some synthetic molecule that we can make. So that's the game. But you can see the game if we want to try to differentiate between cysteine and homocysteine and glutathione. You can see how problematic that might be because that group is very similar. Okay, so how do we differentiate between these systems? And we kind of invoke the use of the chalcogen, sulfur, selenium, and tellurium in some cases. If you have a biological background, a biology background, I wouldn't presume that I know more than you do about how important sulfur is in biology. There's a lot of biomolecules. We're still learning more. And there's selenium is actually an essential trace element. Tellurium is not really, but it's tolerated. But selenium is necessary. So, but the interesting thing with selenium is too much, too little. It's toxic. Just a kind of visual on when we want to make a sensor, we want a kind of strong turn-on event. We want reversibility. We want to be able to have some control about what's going between the different compartments in biology, if we can find out anything about that. So we targeted a natural product that we then derivatized with selenium. This is mycophenolic acid, and it's used commercially as an inhibitor for inosine monophosphate dehydrogenase. So we looked at this system. I guess I'll just say briefly, we made the actual system through a different synthesis, but then we also had a chance to derivatize to something that's fluorescent. So taking a natural product, making a derivative, making something similar that's fluorescent, and so the bromine derivative on the bottom is showing you something that we can work with here. So if we attach a phenylselenide group to this, we can actually have something that is a fluorogenic system. So what I mean by that is when you actually oxidize the selenium, that's sort of like taking away the lone pairs from their ability to meddle in the pi delocalized system, then you can have a strong turn-on effect, a turn-on event. So you can go from selenium, which is divalent, to a selenium, which is tetravalent. And on the left, in the middle, on the left, you can see how there's like a steady signal with the green structure. That's where we have like the nitrogen version of this this phthalate. If we keep the phthalate, that's with the oxygen that's present. That's sort of in the middle, under the heading in vitro study. That's like the little structure that you see there. That actually also does have a turn-on event, but then it decomposes over time. So we think that the phthalate group, that portion, is going to just decompose, just hydrolyze to something else over in short order. But it's also telling us something about biology. So this system was fun to work with. You can see the selenium, right, is present on the arrow group. We were able to look at cellular studies, and in comparison with another group, another system that we also prepared in our laboratory, that is actually a Biophiles system. So the MES-BOD SEPH, that was something we prepared. So I think that was something that Tesla was involved with. And screening, if we think about some ability to determine glutathione in biology, I want to show you this system on the right side, which is a coumarin, as a phenylselenide group that is displaced by a nucleophile. And the nucleophile that we were testing were the Biophiles, and we found that this GSH has a good selectivity for this this probe. So when GSH is introduced, then you have a strong fluorescence increase, and not only is the SEPH group lost to the system, but you also have the involvement of the CHO group. Look at the bottom. If we think about a model system study, which we did, then the CHO group is interacting with the terminal amino group. So this, we think, is the essence of the ability for this probe to detect GSH. So this was a recent research finding in our laboratory. And you can do cellular studies as well. To determine whether we're actually able to get into the cell, and then determine the GSH concentration or the presence in the cell. Okay, so that's the end of my talk. I'm at 48 minutes or so. I thank you so much for hanging in there, and now I want to entertain questions.
Okay, thank you, Professor Churchill. So for anyone who want to ask a question, please raise your hand. And also we have two questions here, Professor, from Mr. Suhartana and Dr. Astuti. Have you ever compared between porphyrin, coral, and choline in terms of reactivity? Which one is more reactive in terms of synthetic point of view? And then have you ever also been compared between your molecules as a photocatalyst with regard to other photocatalysts such as metalloxide?
Okay, so here's like a comparative slide. So I guess for reactivity, the Carol positions at 2 and 3 and 18 and 17 will be relatively reactive. And the other ones will be sort of the same as what you might expect for porphyrin. But the reactivity does sort of depend on the central element, and it depends on steric effects, and it'll also depend on whether the arrow groups are quite electron withdrawing or more electron donating. Let me just say that the Carol is quite an electron-rich system. So you got to try to, I think the game with the Carol is you have to try to remove electron density. So if you're thinking about electrophilic, yeah, it's kind of an interesting question. There's the nucleophilic attack, electrophilic attack, or you could have a radical radical mechanisms. So yeah, I'd say generally speaking, the Carol beta positions are more reactive than porphyrin, but it's gonna sort of depend in some cases what is the central element, and what other factors there are. And the second question about have we compared the photocatalysis with I'm sorry, say that again, with other
like metal oxide, like zinc oxide or titanium dioxide, for example, as a photocatalyst.
Oh, other photocatalysts that are more
Yeah.
kind of like nanoparticle maybe in nature, or
Yes.
so we're trying to kind of make actually, let me just say this: we're trying to make sense out of derivatization with Carols. We don't normally in this sort of chapter I showed you, we're not really comparing over to some completely different system. That's typically what we do. We just try to look at the current derivatization and go step by step. But yeah, that's a good point. We should try to calibrate with very different systems and try to see how they compare. So we haven't done a lot of that.
Okay. Okay, thank you.
Thank
Now
you.
for the next question from Asidara about the relationship of deformation of the complex with its catalytic activity. Could you please try to re-explain to make it a clearer understanding?
Yeah, sure. Well, I guess it's an electronic game, right? So if you have a chance to do some substitution, we're trying to substitute as much as we can. We only were able to get about 5 CF3 groups onto the system, and then and you can see that in the bottom right corner here. So that was kind of the maximum amount of CF3 groups that we were able to put on with our effort at that time. And then you're kind of wondering, well, what does it mean? What is this capable of doing? So then I would say, then let's go over to an energy diagram. And I think it's not very intuitive to work with the HOMO-LUMO levels, but what we were able to do is to just look at all of these related systems and just look at where the absolute levels are, where we, as you're adding, and this was done by, again, my colleague, but we were all looking at it and kind of very interested in the findings. As you go down from the ones to then the 2, 3, 4, 5, you're kind of lowering the absolute orbital energy of the HOMOs and the LUMOs together. So you're making it easier to accept an electron, but you're also, it seems, yeah, it's kind of like a game about whether you're making it easier to accept an electron or for it to lose an electron. So that's what we kind of are still trying to better understand. Is it just the 5 CF3 groups that are doing this, or is it the fact that the ring, the whole macrocyclic ring, is sort of deformed, and that deformation is involved in this change as well? And the deformation comes from the fact that the CF3 groups are kind of colliding with one another, and the fact that they're colliding with one another makes it kind of harder to synthesize in the first place. So you have these kind of interrelated issues about electronics, but basically we want to derivatize to kind of achieve nice kind of electronics and another bottom line was to use inexpensive ingredients. Instead of using gold, we can just use phosphorus, right? So that's a much less expensive element. So we kind of limited ourselves to in this paper to inexpensive elements, and then we tried to make sense out of the electronics. So, but you see a trend of decreasing energy. So this is sort of what we're getting. And it just takes a little while for the students to, I think, appreciate what this means.
Okay, thank you, Professor Churchill. And now we have Dr. Darmawan who raised his hand. So time is yours, Pak Adi.
Thank you. Very nice talk.
Oh, thank you.
I actually I have 4 questions. First, that you are making triplet photosynthesizer using non-metallics as a center. What is the benefit if you compare to the metals? Because usually like we are using metal for the for complex between metal and porphyrins, like this one. This is the first question. The second one, that you mentioned that you are using CF3, and there is a very close correlation between the concentration of CF3 and the reduction potential. So how can you explain the mechanism? Because the correlation between the CF3 and the reduction potential. The third question about the chemical stability of this complex. Is it stable or not? And the fourth one is about the chemical series, because you mentioned or you explained and you saw that there is like homo-lumo, and what's influence the spectrochemical series, because it's like the gap between homo and lumo is become bigger, and what makes the gap between the homo and lumo is become bigger. Okay, that's my question.
Okay, yeah, thank you very much for your questions. So I guess the first question you were asking was, why do we use phosphorus? Why don't we use something like iridium or something? I think our idea was that it's more, it's just more cost effective if we can find a way to synthesize compounds that have, you know, light elements and or like earth abundant elements. I guess it's kind of earth abundant elements. Now that doesn't mean that we're going to wind up with something that's the world's best photosensitizer. There are if you do a lot of derivatization, you can get some room temperature, like, you know, emitter that's you know, that's a phosphor. If you see phosphorescence, right, then you've achieved this kind of this triplet, like this intersystem crossing event. So I guess we were exploring in a kind of one directional way what the CF3 derivatization can do for us in the beta positions. But there's a lot of other directions. But actually, yeah, to try to answer your question bluntly, we were just thinking about an inexpensive and small element that can be ready to use, and that kind of and that kind of relates to another question you said about stability. Yeah, the PF2 unit is, from my experience, is really stable. Once you make a corol, you know, you have to kind of like, you can set like a stopwatch. And if you expose it to air or something, it's gonna start to decompose. Unfortunately, that's the reality. But if you have it as a phosphorus derivative, it seems to have an astonishingly long lifetime. And as I was saying, with the corols, they tend to be electron rich. So the way I think about it is the PF2 unit, for example, inside the axial P's and the phosphorus PF2, that's a kind of internal electron withdrawing group. That's how I see it, and it is very stable. Why is it so stable? It's not going to hydrolyze. If you have the OHs instead of fluorines, if you have the OHs like POH2, POH, POHOH, that will kind of go between a doubly oxidized, a doubly bonded O or two singly bonded hydroxides. It'll kind of have this hydrolytic shift. So, but aside from that hydrolysis, we find it very stable and useful, and also the NMR is helpful. And you were asking about the energy levels.
Yeah, the spectrochemical series.
Yeah. Okay. So when I say spectrochemical series, I usually think of that very simple series where you have a ligand directly bonded to a metal. And if you remember from the inorganic textbook, right, you have the CO and cyano, CN minus ligands having the strongest effect, right? And then you have other ligands. It goes down a list, but the spectrochemical series is helping us rationalize whether we would have a high spin or a low spin. That means like a high spin would be weak field, or a low spin, or that'd be strong field case in simple coordination chemistry. I don't know if that really applies with these macrocyclic ligands directly, but yeah, we can make a case for trying to think of the macrocycle as kind of having more and more electron withdrawing ability, and therefore you would place it somewhere along this so-called spectrochemical series. Yeah. So, but then again, the spectrochemical series is related to the D transition, right? So the D electrons formally, but yeah. So I'd have to think about that question a bit more, but obviously what you find is whenever you add a bromine, you're going to kind of redshift the system. So that's like a given. But then if you add a bromine, where are you going to add it, and what effect does that have? So there's these subtle differences. Yeah. So now I'm trying to think of your other question. Maybe you can ask another question now. I don't want to talk too much.
Okay. Yeah. Thank you.
Oh, thank you. Yeah.
Okay. Thank you, Professor Churchill, for your very interesting discussion. And to all audience, due to the time constraint, so we will
Masih ada pertanyaan?
end up our conversation. Maybe if you have any question, then is it possible to send directly to your email, Professor Churchill, for audience who want to ask for more knowledge and information?
Yeah, absolutely. I always encourage anybody, especially students, to just ask questions, email me. So thank you. It's not, I wish I could see all of you guys, but it'll have to be some other time.
Okay. And so to all audience, please kindly give a big applause for Professor Churchill for his excellent talk today. And then before we close this session, I would like to ask all of you to turn on your video, and then we will have like a online photo session together.
Okay.
Is there a photo? Maybe it's.
And
oh, here's the.
And Professor Churchill, could you please kind to close your sharing desktop, Professor?
Yeah. I'll just I'll let me. Oh, hello. Yeah, I can see you now. Let me just close. Yes, I'm stopping sharing. Is it done now?
Yes, it's done now. So please, for all audience, to turn on your video, and we will have online photo session together today. Okay, I'll start. 3, 2, and 1. 3, 2, and 1, and once more. 3, 2, and one. Okay, so once again, thank you for Professor Churchill who gave a very excellent talk today, and all audience for your attention. And we will see you for the next topic on JKSA webinar series. Aduh kalam, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you for joining.
Thank you very much.
Waalaikumsalam
Thank
warahmatullahi
you.
wabarakatuh.
Thank you.
Oke,
Thank
sip,
you.
sip,
Thank you.
sip.
Thank you.
Waalaikumsalam.
It's done.